Let's look at classification of cytology. It's a big presentation because it's incredibly important. And the outward goal in the end is for you as an RVT to get a slide in front of you, be told where it was taken from, how it was collected, how it was prepared, be able to look at it once it's stained, and identify what the heck are you seeing. Are you seeing cancer cells? Are you seeing inflammatory cells? Are you seeing infection? And be able to report those findings back to the veterinarian for appropriate treatment. So it's really important, hence it's quite in detail and in depth. This is a chart that I want you to get to know most definitely, and these are the classifications that we're going to put each sample into. Sometimes they overlap. We'll talk about that a little bit more down the road as to not overwhelm, but these are the 4 headings that we're going to classify samples into. And then we'll subclassify. So we have infection, inflammation, neoplasia, and non-neoplastic, non-inflammatory. So last week we talked about ears and skin. So don't pigeonhole yourself. Ears and skin don't always belong to this category, but we often see good examples in ears and skin. So the first category, once we've identified the sample as abnormal, is infection. And infection is an absence of white blood cells, but a presence of microorganisms. So looking at that, if we think back to seeing those ear swabs that were strictly yeast, no white blood cells, and or strictly bacteria, no white blood cells, or maybe there are some parasites like ear mites, no white blood cells, those would be classified as infection. So we would get those samples in front of us and we would say infection and then subclassify as parasitic, bacterial, or mycotic, or fungal. Second to that, you should always support your findings. So I'm going to say, yes, it's abnormal, it's an infection, looks like mycotic infection, and then I'll support my findings by saying I saw 2+ yeast, or I saw 6 ear mites, or blah, 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 and no white blood cells. Just to confirm that, yes, I did look for those things, and this is what I found. So here's an ear swab, revealed 2+ yeast, we'll pretend. And how would we classify it? There were no white blood cells, no other findings, no obvious bacteria. It would be infection, mycotic, and then you would support your findings by identifying what you've seen. This ear swab revealed 3+ bacteria, rods and cocci. How would you classify it? It would be infection, septic, or septic infection, either or. Beautiful skin swab of a nice little skin fold would be ++ bacteria and 1+ yeast. How would we classify this one? Because it's not uncommon, especially ear skin, to have lots of yeast and bacteria present. So this one would be septic, mycotic, infection. Or mycotic and septic, doesn't matter, as long as you're noting both and that it is infection. The game changer is when the body starts getting involved and the body starts fighting back. So inflammation is when the body starts sending out white blood cells to help fight against those microorganisms, kill them, and get them the heck out of the body. So inflammation is where we really bump it up a notch and get a little bit more complicated with our classifications and, of course, subclassifications and even subclassifications. So in this case, we're seeing white blood cells. We're going to call it an inflammatory or inflammation sample. And then we have to start looking at the population of inflammatory cells that we're seeing in front of us. With that population, we're going to insert that sample into one of 5 categories. Purulent refers to neutrophils. Pyogranulomatous refers to newts and macrophages. Granulomatous, macrophages. Eosinophilic, thankfully, self-explanatory, eosinophils, likewise with lymphocytic. And lymphocytic is also often called lymphocytic plasmacytic, and that's because of the presence of plasma cells, which you typically see together, especially with reactive inflammation, reactive lymphocytes. So we'll go through each specific category in detail, and then just note this chart, I also want you to take these subclassifications and also attach them to Pyogranulomatous. Okay, just also not only purulent but Pyogranulomatous as well. The 3 steps to classification of inflammation. First, identify the sample as inflammatory. So you might be scrolling through, let's say it's an ear swab, and you're seeing lots of bacteria, lots of yeast, and all of a sudden you're coming across lots of white blood cells. So the presence of those white blood cells means that the body's fighting back, trying to get rid of foreign intruders, invaders, and it's becoming an inflammatory response. Second, classify your sample into one of the 5 forms of inflammation, which we just had on the page prior. Then subclassify appropriately. All right, so step 1, abnormal, it's inflammatory. Step 2, classify into one of these 5 categories. And yes, you need to know how to spell these categories. So let's look at purulent inflammation. Now, purulent, in one way, we've heard of purulent when we're describing bodily discharge. So pus type discharge, white, thick discharge, we often refer to as purulent. That's using the term purulent in a descriptive form. In the way we're going to talk about it now is more of a noun, I suppose, and it's in regard to a specific type of inflammation. So it is purulent, meaning the noun, that's referring to a neutrophilic type of inflammation. So purulent has mostly neutrophils in it. It is an acute inflammation, remembering that neutrophils, once they exit out of the peripheral blood flow, they tend not to last in the body and the tissues for more than 4 days. So they come out, they fight for 1 to 4 days, and then you can start to see sort of not regeneration, but output of other neutrophils to continue the battle. But you also have to assess how well these neutrophils are battling the infection, or how well they're battling in general, and we'll talk about that. So an acute purulent inflammation has 70% or more neutrophils as the population of inflammatory cells. The rest of the cells will be inflammatory, so macrophages, maybe some lymphocytes, pretty rare, might be there, maybe some eosinophils, so various populations, mostly just macrophages, etc. Lots of causes for purulent inflammation. The most common cause is bacteria. So in lab, when I'm bugging you to find the bacteria, find it again, find it in a smaller sample, find it again, it's because if bacteria is the cause of inflammation, bacteria is what needs to be treated. You can get inflammation, especially, any inflammation, you can get without bacteria. So if that's the case, it's a completely different treatment than if it were a septic inflammation, so if bacteria is present. And bacteria, we all know, is quite small, so you have to do a really good job of finding the bacteria in these samples. So this is a great example of a purulent inflammation, and next we'll talk about subclassifying purulent inflammation, or not next, down the road in this PowerPoint. So for full classification of this sample, this would be a septic, degenerate, purulent inflammation, meaning more than 70% neutrophils. They look horrible, so they're undergoing various stages of degeneration. Okay, if you can picture, that one has the the nucleus toward one end of its cytoplasm, same as that guy, and lots of bacteria. Here's another example of purulent inflammation, and in this one you can see a couple of what looks like macrophages, sort of basophilic mononuclear cells, and lots of various material in the background, proteinaceous type strands in the background, fibrin strands, etc. So this is an example of purulent inflammation. Step 2, we're continuing. Next up we have granulomatous inflammation, and that is macrophagic, also referred to as chronic. And it can be sudden onset during very specific processes, such as tissue necrosis and mycobacterium species. But in general, we already learned that macrophages are sort of the second line of defense. So they come out once the neutrophils are dead and dying, when neutrophils aren't really doing their job overly well. That's when macrophages typically come out to help fight. So just note that in very specific cases, tissue necrosis, mycobacterium, you can see macrophages more as the first line of defense, which is pretty rare. Also, FIP, feline infectious peritonitis. I don't know why that took me so long to remember. FIP also is often associated with a granulomatous inflammation or pyogranulomatous inflammation. And just to note that FIP is an evil little virus that cats get, especially outdoor young cats get, and it results in fluid, i.e. pus sometimes, but fluid in the abdomen or fluid in the thoracic cavity around the lungs. And that fluid often contains a high number of macrophages because the FIP virus, well, the virus associated with FIP, actually uses macrophages to further spread the virus. So it gets into the body, it causes an inflammatory response, and then it hops into those macrophages and continues to spread the virus, furthering an inflammatory response, essentially overtaking the entire body. So it's a really, I think it's a super cool virus. It utilizes the inflammatory response to further replicate, but it's pretty detrimental to cats, and it's unfortunate to see. That is a total side note, but important to know. So in these, you typically see just macrophages, mononuclear cells, or sort of those monocytic type cells. And classification would be if it's 50% or more macrophages in your inflammatory sample. Okay, so then we also have macrophages phagocytizing erythrocytes, and we get hematoidin and hemosiderin. Hematoidin and hemosiderin often seen in red blood cell breakdown. Hematoidin crystals. Here's a hematoidin crystal. And then hemosiderin can look orangey brown, could also look a little bit black as well. And I should have fixed this before I did the video, I completely forgot. So it should be erythrophagocytosis, which is macrophage eating red blood cells. That's when you often see hematoidin and hemosiderin. And then technically, I believe it's still hemophagocytosis. So I put leuko for some reason in this case, but it's hemophagocytosis. And that's the macrophage eating white blood cells. Okay, good example here, erythrophagocytosis. Hemosiderin granules. Next up, we've got Pyogranulomatous. So we are referring to two types of inflammation combining. Mixed inflammation. It's a mixture of chronic and active inflammation, and it is typically high population of neutrophils and macrophages. So you can also see giant cells and epithelioid cells. Your qualification for this to be a Pyogranulomatous inflammation is when it's less than 70% less than 70% neutrophils and 15 to 50% macrophages. Causes are the same as granulomatous inflammation, or technically it could be same as purulent when the purulence is when it's transferring over into more of a chronic inflammation. So this is one of my fave samples. This is a good Pyogranulomatous inflammation. We've got heaps of macrophages and then heaps of neutrophils as well. And you can see again, just looking, if you remember me doing that weird body position whereby I'm representing the nucleus going toward the end of the cytoplasm. I'm even doing it now at my computer and you can't see me, which is kind of weird. So lots of degeneration of the neutrophils here. They're not looking like they're doing a good job battling. Okay, small things to point out so that we can come back to them later. Next up, eosinophilic. Eosinophilic's great. It's really straightforward. Often common with parasitic reactions, allergic reactions. Eosinophils tip the scale into an eosinophilic reaction, inflammation when there are 10 to 20% eosinophils. So even if there are 70% neutrophils, if there are 85% neutrophils, if there's 10 to 20% neutrophils, we classify it as eosinophilic inflammation. We would, of course, note the population of inflammatory cells in the background to support our findings. As mentioned in a previous lecture, always be aware if it's a random eosinophilic inflammation for no good reason. Keep in mind mast cells and mast cell tumors somewhere in the body because, again, the mast cell tumors release their histamine granules, which can elicit an inflammatory or an allergic response, and hence the eosinophils come and dump their antihistamines into the area to reduce the problem. But always just keep mast cells in the back of your head. Not physically, don't put them there, but keep them in mind when you're going through eosinophilic inflammation because sometimes they can pop up without realizing it. Also common is eosinophilic granuloma complex in cats especially, and we've seen this in our clinic at the school in a little kitty, where oftentimes they come in and they just look like they have sort of a cartoon-like bottom lip, so a little bit of a swollen bottom lip, or they can get this sort of mustache of eosinophilic granuloma. They also can get it commonly at the commissure of their lips, so right where their two lips meet. And it's an inflammatory reaction. I believe most still treat it with prednisone. Downfall with this one, it can be treated, which is fabulous. It also can spread to the inside of their mouth, and that's what happened to a kitty that we had, who also happened to be immune compromised. He had FIV, but it can spread to the inside of their mouth, and it again can be treated. They can do just fine, but you have to watch out because as soon as you start getting ulcerations in the inside of their mouth and around their mouth, you can imagine that the cat isn't going to want to eat very much. So treatment can actually become quite extensive to treat the symptoms of the eosinophilic granuloma complex associated especially with not eating and cats not eating. So for these, you could do an impression smear on a lesion like that. That one you'd probably do a nice soft swab with some sterile saline and you'd see an eosinophilic inflammation. That's just a picture of steroids, common usage. So here's an eosinophilic inflammation. It's not stained overly well, this slide, but it's still eosinophils, and then sort of a monocytic type cell, macrophage here. Okay, lastly we've got lymphocytic inflammation. Again, it's fairly self-explanatory, not overly common to see lymphocytic inflammation. And with lymphocytic inflammation, it's also often called lymphocytic plasmacytic. You get plasma cells in a lymphocytic inflammation commonly, especially if it's a reactive type lymphocyte that you're seeing quite often, then you'll get a response from plasma cells to come out onto the scene. Chronic local irritation, you can also see it in peritonitis. It's not terribly uncommon because a lot of the plasma cells, lymphocytes live within the villi of the small intestine. And you'll see varying numbers of mature lymphocytes, plasma cells, and you can see Mott cells in a lymphocytic inflammation as well. Again, lymphocytic is like eosinophilic in that it tips the scales at 10 to 20%. So if you're seeing 10 to 20% in comparison to your inflammatory cells, you're very likely looking at a lymphocytic inflammation. Always be sure to assess for malignant changes. So one of the common causes of a lymphocytic inflammation is a vaccine site reaction, and they often just look like a little red bump or a little bump on the skin where a vaccine might have been given. Common, not that big of a deal sometimes. What also can look like that is a cutaneous lymphoma, and cutaneous lymphoma is obviously much more involved than a vaccine site reaction. So you want to look for You could look for lymphoblasts in regard to the lymphosarcoma, but criteria of malignancy, so mitotic changes, pleomorphism, etc. And we'll talk about that more when we talk about neoplasia, but never, ever, ever let that escape your head, that if you're seeing a lot of lymphocytes, and especially a lot of different lymphocytes or abnormal looking lymphocytes, or you're seeing lots of lymphocytes without seeing plasma cells, then always, keep in the back of your head a lymphoma. Cutaneous lymphoma. And again, we don't diagnose it. It's just us giving the information to the vet or sending out our description with the cytology for it to go and be confirmed at the lab. This is just a cross-section. This is human of the small intestine, the little lymphatic lacteals in the small intestine where the plasma cells live and are responsible for immune reactions. So here's an example of a vaccine site reaction. Okay, and here's a good lymphocytic inflammation. Plasma cells and some smaller lymphocytes. Same with here, we've got some neutrophils, some plasma cells, and some lymphocytes. Alright, so step 3, we have to subclassify. So if it was too easy, going, what we've done so far, I'm sure it was much too easy, we're just going to bump it up a notch and subclassify further. So the two categories that we subclassify for are purulent inflammation and pyogranulomatous inflammation, mainly because the neutrophils have such an important role, or the neutrophils are of interest mostly. Sorry, my word's getting messed up as usual. So when we get a purulent or pyogranulomatous inflammation, we want to subclassify it and note, are the neutrophils degenerate or non-degenerate? And are you seeing sepsis, mycotic figures, mycotic, I shouldn't say it like that, mycotic organisms, or parasites? So degeneration, various forms of degeneration that you would note when you support your findings. Karyolysis, and karyolysis is one of the most difficult for an untrained eye to get to know. It's essentially when the nucleus of the neutrophil is starting to stretch out, it's starting to thin out, starting to lose its shape and size in general. The cell itself can actually become swollen. And essentially, end-stage karyolysis, that little nucleus is going to stick itself to the side of the cytoplasm and eventually be disintegrated. Pycnosis is... Oh, sorry, I was just reading that again. But pycnosis is when the neutrophil's nucleus, instead of having that funky fun S shape, has curled into one small concise ball. And karyorrhexis is when it has split apart into various little balls or fragments. Not degeneration is hypersegmentation. Okay, it's a normal stage of aging. If the color is normal, the cell's aged completely naturally. So this is a brief description. Karyolysis is nuclear fading, so it tends to get stretched out, a lot thinner. Pycnosis is nuclear shrinkage. Karyorrhexis is nuclear fragmentation. It ends up being nuclear dissolution and then anuclear necrotic cells. This is an example of a neutrophil that has a pycnotic nucleus. These are neutrophils undergoing karyorrhexis. And in this case, this neutrophil here is karyolysis. Okay, and then this is end-stage karyolysis. So this is really a moth-eaten appearance. The nucleus is fading. It's becoming very, very thin, very stretched out. And you can see here even the nucleus is shifting to one side. So that's karyolysis. And karyolysis has so many different stages, it can be really difficult to tell at first. These are just hypersegmented. Okay, so just a little recap. What is degenerate versus non-degenerate? And always keep in mind your sample has to be well smeared and stained to view accurate changes. You don't want to have a lot of smushed cells, so basket cells, smudge cells. Always look for an intact cytoplasm as well. Okay, further, so again, just attach these three little categories also to Pyogranulomatous. Further, we're going to subclassify it as sort of what is causing this in the purulent and Pyogranulomatous. So we'll subclassify purulent and Pyogranulomatous as degenerate or non-degenerate. And then we're going to say, is it septic or non-septic? So septic, as an umbrella term, refers to microorganisms present. Septic as a descriptive term, well, it's kind of both descriptive, but septic in specifics, we're going to refer to it as, is there bacteria present? So we will talk about septic as bacteria from here on out. So ways that we can subclassify purulent and Pyogranulomatous inflammation is to further identify if it was parasitic, septic, or mycotic or fungal. And common types of infection in inflammation would be parasitic. Often you might see, well, not often, but you might see Diarrhea imidis. Septic, so bacteria, Staphylococcus, Pseudomonas, Aspergillus. Mycotic, Blastomyces, Malassezia, Cryptococcus. Okay, so there's blasto. Not uncommon to see on a dog with blasto if you do transtracheal wash or bronchoalveolar lavage. Diarrhea imidis in an inflammatory sample. Alright, so let's recap everything. Step 1, identify as abnormal, and then classify it as either infection, inflammatory, neoplastic, non-neoplastic, non-inflammatory. Step 2, we've decided it's inflammation. So we have to identify, is it purulent, pyo, granulomatous, eosinophilic, or lymphocytic? Step 3, subclassify. So subclassify it as degenerate or non-degenerate, septic, mycotic, parasitic, or non-septic. Always note what's seen. So in test quizzes I'll prompt you, I'll say classify this sample, support your findings. So full supporting your findings, you would say, let's say it's purulent, septic, degenerate, inflammation. So you've told me everything I need to know. For supporting your findings, you would say it was more than 70% neutrophils, so therefore it's purulent. It was white blood cells were present, that makes it inflammation. And then I said it was septic because I saw 2+ cocci. Now let's practice. So this is the basic. This is just into the categories of inflammation. So looking at this sample, is it purulent, granulomatous, pyogranulomatous, lymphocytic, or eosinophilic? Having a quick count of our cells, we'll come to the determination that it's purulent inflammation. These are macrophages that are from mucus. They were from, I can't remember specifically these ones, but mucus-type macrophages tend to be very frothy, very foamy looking. These are likely respiratory tract. And I just gave it away, saw a whole bunch of macrophages, so we're going to call it granulomatous, greater than 50% macrophages. So again, looking at our cell population, we've got macrophages, we've got all these little eosinophils. I'm going to call it eosinophilic because it only takes 10 to 20% eosinophils to bump it into eosinophilic. This one's a mixed population, so we've got lots of neutrophils, we've got lots of macrophages. We have to do a full count, or at least count in varied areas, get an understanding of the general population. And with that, you can come to the determination that it's pyogranulomatous inflammation. So again, I'm not sure why I showed this one again. Was I going anywhere with this? Let's see. Yes, we're going to bump it up a notch. That's right. So this one again. So we're going to have a look, and we're going to call it purulent, non-septic. I'm not seeing any bacteria. I'm not seeing any yeast, and non-degenerate. Lots of hypersegmented little neutrophils, okay, or heavily segmented neutrophils. Non-degenerate. Purulent, non-septic, non-degenerate inflammation. Looking at this happy little guy, we've got this beautiful little plasma cell. Lymphocyte, 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 neutrophils. So I think we're going to call it lymphocytic inflammation. Okay, this bad boy, we've got 2 macrophages, and we've got 3 neutrophils. So to classify, pyogranulomatous, and then we'd further classify, is it septic and is it degenerate? Pyogranulomatous, non-septic, non-degenerate inflammation. And then this butte from the very beginning, this is our heaps of neutrophils sample, lots of bacteria, lots of degeneration. These cells are pretty much all karyolytic. Okay, lots of karyolysis going on there. They don't look like neutrophils anymore. So we're going to call it purulent. Have to consider if it's septic or degenerate. So it's, that should say purulent inflammation. Cross that off, should say purulent. So purulent, septic, degenerate inflammation. Always report what you're seeing. Report the classification of samples and your percentage approximately of what you're seeing. And that is cytological classifications with a focus on inflammation.